My name is Bilge O, Zdemir. I'd like to thank the organizing committee for giving me this opportunity. In this presentation, I will talk about our study investigation of the effect of Toll-like Receptor 4 and Fc-gamma Receptor III inhibition on macrophage activity. Let's start with some basic information about the background of our study. Citronation, a post-translational modification, is a physiological process that increases the durability of proteins. The enzyme peptidylarginine deaminase, or PAT, converts the arginine amino acid into sterline. However, in some autoimmune diseases such as rheumatoid arthritis, increased enzyme activity leads to hypercitronation, making proteins immunogenic. Proteins that become immunogenic through citronation activate some autoreactive B cells and cause them to produce autoantibodies called anti-citrullinated protein antibodies, or ACPAs. Another focus of our study is toll-like receptors, and studies have shown that many endogenous proteins like fibronectin, fibrinogen, or tenascin C involved in the pathogenesis of rheumatoid arthritis can lead to TLR4 receptor-mediated immune activation. Why these interactions are important? Because citrullinated peptides, or CPs, in the immune complex with ACPAs bind to Fc receptors on the surface of the macrophage or dendritic cells, and at the same time interact with TLR4, causing dual activation. As a result, a complex immune activation is triggered that it is self-sustaining, involves T cells and fibroblasts like synovial. There is antigen maturation in B cells, inflammatory activation in T cells, and increased release of catalytic enzymes and cytokines that cause cartilage damage in FLS. Macrophages are the center of the interactions of these different immune cells. Therefore, in our project, we aim to examine in detail the mechanism that cause activation in macrophages and to discover potential therapeutic targets. For this purpose, we collect blood samples from rheumatoid arthritis patients with high ACPA levels and in the active disease stage. We use blood samples from age and sex matched healthy donors as controls. We isolated PBMCs from blood samples by picoral density gradient methods. After 7 days of culture with MGSF and interferon gamma cytokines, cells were differentiated into macrophages. In macroscopic examinations, we observed that macrophages of healthy and rheumatoid arthritis patients had slight morphological differences. We evaluated macrophage differentiation by using flow cytometry method with CD14, 32, 64, 86 markers, and confirmed that cells differentiated into macrophage. We created 6 different experimental groups as shown in the figure. In this way, we evaluated the effects of blocking Fc receptor and TLR4 activation induced by ACPA immune complex on macrophage phenotype and secreted cytokines. We used flow cytometry data to observe change in CD14, 32, 64, 86, 200R, and 163 expressions of the groups and compared healthy donors showed blue with rheumatoid arthritis patients showed red. We observed that CD32 and 86 MFI values of macrophages from rheumatoid arthritis patients increased significantly after LPS stimulation, which is the ligand of the TLR4 receptor, but decreased significantly when TLR4 inhibitor was applied. On the other hand, CD14, 64, 163, and 200 RMFI values decreased significantly after LPS stimulation, but increased significantly with TLR inhibitor application, except for CD64. CD14, 32, 64 MFI values of rheumatoid arthritis macrophage stimulated with ACPA immune complex decreased significantly in the group where TLR4 and Fc inhibitors were administered together. Interestingly, this difference was also significantly lower than TLR4 inhibitor and Fc inhibitors administered alone. On the contrary, after ACPA immune complex stimulation, CD86, 163, and 200 RMFI values increased significantly in the group where TLR4 and Fc inhibitors were administered together. Again, this increase was significantly higher than the groups administered TLR4 inhibitor and Fc inhibitor alone. We determined by ELISA method how the levels of inflammatory cytokines TNF-alpha, interleukin-1 beta, and interleukin-6 secreted from immune cells and especially macrophages in the pathogenesis of rheumatoid arthritis change in the experimental groups. When we stimulated macrophages from rheumatoid arthritis patients with LPS, we found that TNF-alpha, interleukin-1 beta, and interleukin-6 levels increased significantly, and there was a significant decrease after TLR4 inhibitor application. A similar situation was also seen in health controls cases. TNF-alpha, interleukin-1 beta, and interleukin-6 levels increased significantly after LPS, but TLR4 inhibitor only made a significant decrease in TNF-alpha and interleukin-6 levels. There is no significant change in interleukin-1 beta levels. Citrulated peptides are known to be ligands for TLR4. Therefore, we only examined the effects of peptides of macrophage activation compared to their ACPA immune complex. In our findings, we observed that in rheumatoid arthritis group, ACPA immune complex made a significant increase in TNF-alpha and interleukin-6 levels compared to peptides alone, but didn't cause a significant change in interleukin-1 beta levels. Again, in the rheumatoid arthritis group, we found that TNF-alpha and interleukin-6 increase induced by ACPA immune complex were significantly reduced by TLR4 inhibitor alone, Fc inhibitor alone, and their combination, but interleukin-1 beta levels were significantly reduced only with the combined inhibitor. The changes for TNF-alpha in the control group were similar to those in the rheumatoid arthritis group, but inhibitor administration didn't significantly alter interleukin-1 beta and interleukin-6 levels. As a conclusion, both stimulated peptides and their immune complex forms can differentiate macrophages into a pro-inflammatory phenotype. When administered alone, TLR4 or Fc receptor inhibition can reduce the effect of stimulated peptides and ACPA immune complex. However, the dual blockade approach seems to be more effective than TLR4 or Fc receptor inhibition alone in effectively controlling both the macrophage phenotype and the inflammatory cytokines secreted by it. Last, I'd like to thank two other researchers who participated in the study and Jalal Bayar University for supporting us. Thank you.
Thank you so much. That was really interesting. We have some questions. The first one will be from Dr. Fish.
Great talk, thank you. So I noticed you focused on rheumatoid arthritis. I'm just wondering, because there have been a lot of studies comparing RA and osteoarthritis, OA, and looking for inflammatory markers and so on and so forth. Have you considered using osteoarthritis as a control and seeing whether those inflammatory markers still, and the peptides, the citrullinated proteins and ACPA immune complexes are specific to RA?
Yeah. Osteoarthritis is another disease in it, not like rheumatoid arthritis, and we didn't.
Oh, you didn't use that. All right. It's just that, TNF and IL-1 and IL-6 are up in those as well, OA patients as well. So it's always been tricky to try and tease out what's specific to an autoimmune disease compared with just an inflammatory disease. So my next question is, you mentioned macrophages. Do you think other immune cells are important in the context of those citrullinated proteins or the ACPA immune complexes? You mentioned macrophages. Do you think other cells are important, like fibroblasts or B cells?
Sorry, can I interrupt this presentation? I am another researcher in this project. Maybe I can explain
more
better.
Absolutely, go for it.
Is it okay for you? Sorry. Yeah. Actually, you are right. In that perspective, especially B cells plays important role, but also after this immune milieu leads to activation of fibroblasts like synoviasis, and these are important because the main actor of the cartilage damage. They proliferate uncontrolled manner, and then they secreted many cytokines and also and microenvironment catalytic enzymes that caused sort of cartilage damage, and they also express TLR4 and Fc receptors. Therefore, maybe in the next projects, we also just focused on the FLS, fibroblasts like synoviasis. But we attend this meeting from Turkey, and unfortunately, Turkey, the economic situation is not good enough, and we having trouble getting money for performing research. Therefore, maybe a couple of years later, we can explain our future findings with your seminar or conference.
It sounds fantastic. Yes, synovial-like fibroblasts are really important. It's worth having a look at those for sure, and there are a number of models. But thank you very much. That was great. Back to you, Irina.
Thank you so much.